there's a new development. We used to always think that the krill is just feed on the surface. But a couple of years ago, some people from the British Antarctic Survey, they actually had a camera down at 3,000, over 3,000 meter depth, and they found krill feeding. Yeah, that's right. They found krill feeding on the seafloor. And you can also see that this female with all the green stuff in her hepatopancreas is full of eggs. So they were feeding down there and also making eggs. But this we know that the eggs can only be laid at the surface because they're laid at the surface and then they sink down and then they hatch. So the krill has this kind of life cycle where the krill larvae, spend a lot of time at depth where there are much fewer predators than at the surface. So they're escaping predators that way. But the fact that they can do this, or that they have done this, leads me to suspect that they will be doing this all the time. So when the diatom bloom is built up in the surface water and starts sinking, the krill will go down with it, if there are krill there. In our experiments, there were no krill because we went into regions, we had to do the work in regions where there was no krill. So we couldn't follow this. But I'm sure if there were krill in IFEX, that they would have also gone down and started feeding on the diatoms at the bottom and then come up again. So they would have brought the iron back up again. So this would, again, be recycling the iron. This way, the krill was recycling iron.
So to summarize, maybe for our listeners, if we can basically choose by selecting a certain location with a higher population of krill or a lower population or a no krill population, if we were to optimize for carbon export, or if we want to optimize for building krill biomass, and if we want to build krill biomass and whale biomass, and if we want to boost the ocean food web. Yes. So, and if we were to build a model just for the sake of the podcast, and let's say we do select a region of the ocean that has krill feeding on diatoms, do you believe that 100% of the diatoms are being, let's just assume, let's talk a little bit about the depth of the Southern Ocean. So what would happen if it's 3,000 meters deep or 2,000 meters deep? Is there a difference, or just 1,000 meters deep? And is there a difference between krill feeding on diatoms very deep or feeding less deep? And would that possibly influence the permanence of the carbon removal, and also in general, what happens to the carbon consumed by the diatoms, and then the diatoms consumed by the krill, and then the krill consumed by whales and other animals? How can we look at the carbon permanence? Is that carbon being out of the cycle, and if so, for how long? And does it maybe depend on what animal is eating ultimately the krill?
Yeah. Look, you see this photograph, you don't see it very distinctly, but you see a little bit of green fluff, it's called, lying on the surface. And this is a deep sea environment adjacent to a coastal diatom bloom. So this coastal diatom bloom sank out over there. Now, what you asked is, okay, you asked many questions. All right, let me start off by saying that when diatoms start sinking, it's like snow formation. They start building the snowflakes, right? They start growing their biomass. These diatoms die, and I mentioned that previously. They clump together into flakes, and then they sink out. And then it doesn't matter how deep the water column is. It can be 10,000 meters. They reach the bottom because they sink down very fast. It's like when snow starts forming up at high altitude, then that snow, most of that snow comes down. Right? If there's only little snow, then that might evaporate on the way down or something, right? But once the flake is formed, it just goes down and hits the bottom. This is what will happen over here. So the total water depth doesn't matter, whether it's 20 meters like in Kielbeit or 3,700 meters, which is the average over here in the Southern Ocean. That doesn't matter. That stuff is going to land on the seafloor. And once it lands on the seafloor, it's washed by Antarctic deep water, which forms around Antarctica. And that's the water layer that covers the abyssal plain. And that water is going to stay down there for the next 1,000 years. because the Antarctic is the place where this water is formed, freshly formed bottom water, and that has a residence time of 1,000 years. In about 1,000 years, the entire ocean is mixed. That the deep water comes up in 1,000 years. So whatever lands down there is gone. Of course, when the krill now follow it and pick that stuff up, then that iron is then brought back up again, and the carbon with it, of course. But the net amount of iron the net amount of carbon would still be more than if you didn't do it. It would still be sequestering carbon because a lot of that stuff gets buried in the sediment. There are worms living, these are long worms that live in productive regions, and their job is they come out of the sediment and they pick up whatever is lying on the surface and pull it down into their hole, what earthworms also do. So they pull it into the sediment, and then that sediment, so that carbon is gone. And it's gone for geological timescales because that sediment then builds up, and the stuff that they took down is going to stay there. There are many mechanisms by which the particles on the surface are recycled. The different kinds of organisms will grow there and do this kind of thing. But the end effect is that stuff is going to still stay. Even if it comes back into the water, that water is going to stay down there. Right? So this is really long-term sequestration, really long-term sequestration. The reason why diatoms sink to the seafloor is because they make resting cells and take that with them down to the bottom, right? Now, these resting cells then land up in the sediment, and in the long term, it's expected that those cells will be resuspended and they can grow again in the surface. Because by going into the sediments, they are less in danger of being eaten or grazed or being killed by animals. Now, of course, the deep ocean, when diatoms sink out in the deep ocean, they don't, that deep ocean water is not going to come back. It's down there for 1,000 years, right? So that's not going to help them. But this is the situation with overspill in biology in general, like when plants that have pollen that fertilize pine trees, for instance, they may produce pollen, and that pollen is blown by wind, and it then one in I don't know how many millions or trillions reaches a female flower and fertilizes the seed. So this is normal in biology, that a huge amount of investment goes into multiplication or propagation or reproduction, and the outcome is very small. So you have that situation here also. Now, if you start doing this iron fertilization, building up these species, so if they have resting spores in the water that we are fertilizing, then we have a very high efficiency of carbon sinking out, because more of the iron is going into those species. Whereas if you go to the open ocean now, and so anywhere in the open ocean, there will be fewer of these spore-forming species. But you can build up the spore formers, because in the open ocean, they concentrate their biomass in what's known as the pycnocline. This is the layer between the mixed layer at the surface and the deeper ocean. So there, it's like a kind of artificial seabed. Yeah, that's right. That's right. Those are the classic resting spores of species that want to hit the bottom. Exactly. Those we would like to have, and then we increase the efficiency. And the more iron fertilization we do, the more these species will be favored. And the less the other species that want to stay at the surface, that pick up all the nutrients and stay there. And these species, they only shed the silica frustule, meaning the shell. Only the shell sinks out, the carbon stays up there, which is why the sediment is full of diatom shells in the Southern Ocean. There's all diatom shells lying on the seafloor. But there's very little carbon in those shells, because this is mostly those species that don't have a lot of carbon, and their shells survive because they have very thick shells to avoid being grazed. If you want to stay at the surface, you also have to be more protected against being eaten, right? And these are the species that, like behind your head is one of them. Huh? These pennate diatoms that have thick shells. Anyway, so if you get these thin-shelled species, they take the carbon down with them and bury it, particularly in resting spores. And that we've shown that during the glacial periods, when there was much less CO2 in the atmosphere, there was the area on which this spores of Chaetoceros concentrated was much larger. So these Chaetoceros spores, this genus of diatoms called Chaetoceros, this has been cooling the planet in the past. It's been a major vehicle for removing carbon. Yeah, they have these spines. Exactly. Yeah, you've done good work. And they have these long spines, which makes them difficult for copepods to feed on. So if a copepod wants to feed on these diatoms, it has to grab the chain and bite all the spores off because it cannot ingest them. So it takes time. They do that. It's been filmed, right? They grab a thing and then all the like hair, the spines fly around, and then they eat the chain itself, like that, right? But if they didn't have the spores, they would be eating many more at the same time, right? So the handling time is important here. Anyway, the shells of these diatoms, they dissolve. So you don't find the diatoms themselves in the sediment, you only find their resting spores because they survive, right? And they can also go through the guts of copepods, the intestines of copepods, survive and then grow again because they're so tough, right? These are what you now have on the screen are vegetative cells. But in this one, in the bottom one, yeah, that one, the lower one, no, to the right, those are resting spores, exactly. Those are resting spores inside yeah, exactly there. Those are all resting spores. And the other chains on the left, for instance, the three on the left, they have the plasma, the normal plasma with chloroplasts and so on. And they then get broken down. But if the carbon is packed in these resting spores, it's gone for good. And here it's a deep sea sediment, then it's gone for very long. Anyway, so
another question that always comes up in this question about permanence and the rate of carbon export is what other scenarios of decomposition of Chaetoceros exist? Do bacteria eat them, and under what conditions and in what depth do bacteria start decomposing them? And if we were to model it out, does it happen at the top layer of the ocean within the first 20 meters, or does it happen deep down where the carbon might be stuck anyway?
yes, this what you just said applies to those diatoms that die in the surface layer and don't sink, don't make flakes, right? The Chaetoceros, because of these spines, you see these long spines, they aggregate, they become sticky, and they make flakes like snow. And when you have clouds, the clouds are there because you have droplets, water droplets. Those are the clouds. And when those water droplets get big enough, then they start sinking. But when they're very small, they can't sink because they're kept in suspension. So you have white clouds. But once they reach a certain size, then they become heavy. Then they get weight. And then they start sinking. And they then sink down to the seafloor. And those ones sink so rapidly, like in weeks, they reach a 3,000 meter depth before anything can break them down. But the diatoms that stay in the surface and all the other phytoplankton species that want to stay in the surface, they fall apart, they decompose in the surface layer by bacteria and so on, right? And those are the recycling ones. But a certain proportion of these of the organic material formed by these species that want to stay at the surface, what we call silica sinkers, they are decomposed in intermediate depths. And this is where the so-called oxygen minimum zone arises.
Okay, to summarize this point, it depends what phytoplankton is growing. I think that's always important. In any case, we would take a baseline, right? So we would take some water samples and look under the microscope, how much Chaetoceros, how much spores, how many, what algae do we predict will sink out and won't be eaten up, and how many of them will. And it really depends on which location we select.
Yeah,
and
that's important.
The location predicts the efficacy for carbon export. The location predicts how much will be eaten by krill, and the location predicts if there is any that will be eaten up by bacteria, and if this leads to things like anoxic zones. And I think this is one of the big questions, maybe something where we can dive in now a little bit deeper.
As I mentioned to you, there are these different types of plankton, and there are these diatoms, and then there's phaeocystis. So basically, you can say there are two kinds of plankton: the diatoms and the others. And the others are flagellates. Flagellates means that they have a cell with flagellum on it, or more. They can have 4 flagellae in some cases. And so they swim around. And these are just little lumps of plasma. And their enemies love to eat them because it's easy to eat. They have to capture them and then they eat them. So the flagellae mainly there to escape from being eaten. Right? And diatoms don't have that because diatoms, they have a shell. And the shell protects them. So they have no means of locomotion other than when they're on the surface. But in the water, they can't do anything. So they have to rely on their armor to save them. Okay? Now, Phaeocystus is a flagellate which is trying to be a diatom. It has these small cells like all the other flagellates, but it grows a capsule around it, the cell. And this capsule is very tough. We've tested it, and we have taken Phaeocystus colonies. They make colonies, right? So the cell inside this capsule starts dividing, and this capsule goes bigger and bigger and becomes a pretty big colony, bigger than diatoms in some cases, and covered by this balloon-like skin, which is very difficult to puncture. Those places where there's less krill, you have more Phaeocystus because they can then start growing and building up these colonies. And they then start dominating the phytoplankton biomass because they also have high growth rates. Okay, but where there's krill or where there's a lot of zooplankton, then these capsules, they get bitten in the bud, so to speak, right? While they're making capsules, they can still be chomped by the zooplankton. They have very sharp teeth. In the Atlantic region, where you have the krill, you don't have much Phaeocystus blooms. And the reason why Phaeocystus and diatoms are different is because the Phaeocystus has just a layer of balloon-like skin around it, and it doesn't sink the way diatoms sink, because the diatoms are enclosed by this silica, which is also heavy. It's silica frustules. So the whole thing sinks out, whereas in the case of Phaeocystus, it's less. And under certain circumstances, you can have some sinking out of Phaeocystus. Okay? Phaeocystus belongs to this group. Now, the others that I mentioned over here, like Prochlorococcus and the small flagellated algae, these are everywhere. Now, except for, okay, Synechococcus and Prochlorococcus, they're primarily tropical species. You get them in warm water. But all the other flagellates, you get everywhere in the entire ocean. So Phaeocystus is also, there are different species of Phaeocystus, and they're all over the world, in all the oceans, also in the Indian Ocean and in the Arabian Sea. And it's more or less the same species as you have in the North Sea, whereas the Antarctic and Arctic species of Phaeocystus are different, right? But basically, the structure is the same. So what you have is, if you want to visualize what the plankton looks like, you have the surface layer, well, they have published this also, I mean, in essays, easy to understand essays in Nature. It's called The Ocean's Veil, this paper I'm talking about. It's a layer of these small flagellates and bacteria and little ciliates that feed on the flagellates, and they are there permanently. It's a microbial network. It's always there. And when there's nothing else, it's only them, right? So in the ocean's deserts, like in the subtropical gyres, you have these microbial networks which dominate the plankton because there's nothing else. In the Southern Ocean, you also have this microbial network, but on top of the microbial network, you have the diatom blooms. They are superimposed on the microbial network. Right? So the diatoms, they come and they go. So they make up their biomass and then they sink out. But the microbial network stays there all the time. It just gets less in winter because it's being grazed inside itself, right? And this is most of the primary production that people talk about when they say the ocean is producing are these small guys. But they might be dividing like 4 or 5 times in a year or even more. They have division rates of 1 a day about, right? And bacteria also, they're about the same rate of cell division. But the bacteria are also being fed on by flagellates, right? So the bacterial concentrations are constant throughout the ocean. They're at 1 million cells per milliliter in the surface layer, and they're everywhere. It's the same because that's where they're hit by viruses and eaten by flagellates. So they don't count. In the carbon cycle, they don't count because they're just recycling that carbon. They're not building up anything and removing it. Now, in the case of diatoms, they make these blooms, and they take up carbon dioxide from the water, which is then replenished from the atmosphere, and then they sink out. So they make a hole in the carbon layer at the top, whereas the small flagellates don't. They're just recycling communities. That's where the confusion is. And people don't understand that. On land, when something grows and divides, it's there the next day as well, right? When the leaves start forming in spring that come out, the trees around here are all green. And these leaves were formed in April, May, and they're still there. And the oxygen that they gave off is still in the atmosphere. But over winter, they'll sink out, the leaves will fall, land on the surface of the soil, incorporated in the soil, and a certain amount will then be recycled by earthworms and stuff like that, and the soil fauna, and then go back into the atmosphere, the CO2, right? But in the water, that stuff is cycling much faster, right? And this is what people don't get. They don't get that the cell which is dividing today is going to be eaten tomorrow. And that carbon dioxide is released again. So there's a fundamental difference between production in the water, in plankton, and production by space-holding plants. I call that space-holding. Actually, I don't call it. That's the term for it, right? And there are no space holders, real space holders in the plankton, because they have to go and come back again. So the plankton communities in the southern, in anywhere in the plankton, they only last for weeks, right? 4 or 5 weeks is average. And in the tropics, it's less. It's 1 or 2 or 3 weeks. But the populations are being replenished all the time. And it's the same nutrients going on and on. Today they are in this coccus, and tomorrow they're eaten, and it's going into the silicococcus, and then it's going into some other flagellates. And so it's just going round and round. And the rate of recycling has no effect on the carbon cycle, global carbon cycle at all, whether it's going like this or whether it's going like this. It's the same amount of carbon. It's just staying there. Are you understanding now? And that's where the confusion comes. Okay, so there are a range of species that are cosmopolitan. They occur practically all over the oceans. And then there are also species, and the Southern Ocean has the largest percentage of species that are what we call endemic. They only occur in the Southern Ocean. The blooms in the Southern Ocean, because it's much colder, they take much longer. They take 3 to 4 weeks to peak, whereas in the equatorial Pacific, they would take like within 1 week, you would reach the peak, or even less, perhaps. And in the Canadian and in the North Pacific, Canadian and Japanese experiments, there the water is also warmer, and that takes a bit longer, right? A bit longer than the Southern Ocean, but less than in the equatorial Pacific.
So does it also relate to the silica concentration? And if we understand silica as a proxy for diatom dominance?
Yeah. No, that's a very important question. And silica it behaves quite differently to the other nutrients, nitrogen and phosphate, because it's not involved in biomass buildup. It's involved in making frustules, the shells of the diatoms. And it has a different behavior regarding its dissolution. When a silica frustule dissolves, it takes much longer than an organic molecule or organic biomass to dissolve. In the North Pacific, you have very high silicate concentrations in that upwelling water that comes up because it's the end of the conveyor belt. So there would always be a lot of silicate in the North Pacific. In the equator, on the other hand, it's different along the equator because the water upwelling along the equator is fed by Antarctic intermediate water. This is the water this is a water mass formed along the northern rim of the Southern Ocean that subducts under the other oceans. And this water has a lot of nitrate and phosphate, but very little silica, because the silica is taken up by diatoms growing in the Southern Ocean, which I will explain next. All right. So in the equatorial Pacific, for instance, the amount of silica is so low that you can prevent diatom blooms from happening, although there's a lot of nitrate and phosphate. So the HNLC, so there's a story, HNLC, there's now also HSI and CSI, anyway, high and low silica. You have to take that separately. Now, what causes that? This is what we very clearly showed in the Southern Ocean. So in the Southern Ocean, you have this nitrate and phosphate coming up, and you have blooms in places where there's iron availability around islands and from the peninsula and so on. They have big blooms, and they take up nitrate, phosphate, and they also take up silica. But in the rest of the Southern Ocean, the silica is taken up despite the nitrate and phosphate not being taken up. So there's an excess of nitrate and phosphate, the silica is gone. Where does that silica go if there's no nitrate and phosphate? And this is what we showed. This alga, the one that grew, that they found 2 weeks later in Soiree, 4 weeks after the experiment, this Fragilariopsis, this is a diatom which has a very different strategy to those diatoms that make the blooms. This is a diatom that tries to persist in the surface layer. That's its evolutionary strategy. It's been selected for that. It has very thick frustules, very powerful, very strong. And we carried out here from the Alfred Wegener Institute, together with other institutions in the Helmholtz community, we carried out micro manipulation and experiments with shells. We tested the strength. And we found that this Fragilariopsis shells, the strength of the shell is so strong that if you were to take it up to table size for us to relate to, you could have an elephant stand on one of these shells, just to give you an impression of how strong they are. So this is of great interest to biocomposite people because the strength comes from elasticity. I can talk for hours about this aspect, the material properties. Anyway, so this shell is extremely strong. It's made to be very strong. And what happens is that this particular species is so strong that it's also swallowed by copepods whole. And it goes through the gut and can also emerge at the end and come out with the feces, and it's alive. So it survives gut passage. Not all, the majority do get crushed. By the way, a paper just came out now. This interests me a lot. I'm very happy to see it. These are Danish colleagues with whom I've been interacting. And they showed that the teeth of these copepods have teeth like that, right? With which they crush these diatom shells. And they showed that there were copepods that they fed with diatoms and then with other organisms, like the one behind your shoulder, that Dinophysis. And the ones that fed on Dinophysis, their teeth were intact. And the ones that fed on these heavily silicified diatoms, they had broken their teeth. You clearly see that there were pieces broken off. So for them, it's the level at which they can crack shells is reached over there, right? And you all know there's a saying that you don't look a gift horse in the mouth. You also have that in German. Why? Because the teeth get ground down by the silica in the grass. So grasses protect themselves also with silica, just like diatoms. And they just have pieces of silica. They don't have a shell out of silica. They just have pieces of silica in the grass. And that's where you cut yourself on. You can cut yourself on grass. I'm sure you have. And particularly this, what we call pampas grass, is very tough. And that's because of silica. That silica protects the organic matter, and it wears down the teeth of herbivores, including horses. Right? That's why horses become old, because their teeth are down. And the cows and horses and so on, they only live for about 20, 25 years because their teeth are ground down by that time.